الله الرحمن الرحيم. Assalamu alaikum wa rahmatullah ta'ala wa barakatuh. I would like to thank all members in American Central Conference for giving me this chance to present my paper titled Human Monkeypox Virus Zoonosis Disease. This review paper has been accepted in Journal Science (JOES) journal. I'm Rihab Dafallah, Assistant Professor in University of Gezira, Faculty of Science, Department of Microbiology, Sudan. Introduction. Human monkeypox is a zoonotic viral disease which resembles smallpox clinically but differs from it epidemiologically. It is an endemic disease in Western and Central Africa, where there are equatorial rainforests, and the animals that may contain the virus completely live. Monkeypox chiefly occurs in communities where there is often a high background prevalence of malnutrition, parasitic infection, and other significant health compromising conditions, any of which could impact the prognosis of a patient with monkeypox virus. The major outbreaks in 1996 and 1997 was in the Democratic Republic of Congo. Monkeypox is endemic in these 11 countries: Cameroon, Central African Republic, Republic of Congo, Gabon, Cote d'Ivoire, Liberia, Nigeria, Sierra Leone, Democratic Republic of Congo, South Sudan, Benin. Globally, prior to the 2022 outbreak, mainly reported in Central and Western Africa, globally outbreaks are due to exotic pet trade, raw meat consumption, and international travel, increasing its potential to become a global pandemic. More than 23,000 cases in countries that have not historically reported monkeypox. In US, about 60,000 cases, all states except Montana and Wyoming. In northern countries, 60 cases in 15 countries. The Fisheries table shows countries that hasn't historically reported monkeypox. The maximum about 23,000 cases reported in United States, while the minimum about 1,000 cases reported in Colombia. Second table shows countries that historically reported monkeypox. The maximum about 300 cases reported in Nigeria, while the minimum about 3 cases reported in Congo. History of the disease. The disease is called monkeypox because it was first identified in colonies of monkeys kept for research in 1958 after 2 outbreaks of a pox-like disease in them. It was only later detected in humans in 1st of December 1970. First human case diagnosed in a 9-month-old baby boy admitted to the Basankaso Hospital in Zaire, now the Democratic Republic of the Congo, during the intensive search for smallpox cases after its elimination in 1968. In June 2003, the first condition of monkeypox virus in the Western Hemisphere was in the US. Historically, evidence research travel to endemic areas, interaction with wild animals imported from endemic areas, and providing care to an infected animal. Causative agents. The monkeypox virus belongs to the family Poxviridae, subfamily called Poxvirinae, genus Orthopoxvirus, species monkeypox virus. Other important viruses in the Orthopoxvirus genus are variola virus, smallpox, vaccinia virus, smallpox vaccine, cowpox. Monkeypox virus disease has symptoms similar to those of smallpox, but with less severity. Monkeypox virus is large, 200 to 250 nanometers under electron microscope. brick-shaped and surrounded by a lipoprotein envelope with a liner double-stranded DNA genome, rely on host cell ribosome for messenger RNA translation. Poxviruses include all necessary replication, transcription, assembly, and egress proteins in their genome. Transmission. Zoonotic transmission, animal to human by direct contact with animal. Blood, bodily fluids, cutaneous skin-related, or mucosal lesion of infected animals. The viral infection has found in animals like squirrels, rats, termites, and different species of monkeys that believes as reservoirs. Eating undercooked meat of infected animals is a risk factor of infection. Human-to-human transmission can occur by contact with respiratory secretions. From face-to-face contact, skin lesion of an infected and contaminated objects. Transmission can also occur through the placenta from mother to fetus, congenital monkeypox, or proximity after birth with an infected person. This slide shows picture of most animals that live as reservoirs. Monkeypox transmission cycle in Central Africa. Primary transmission from primary host and incidental host to bushmeat hunting and transmission to humans. Secondary transmission from humans to humans. Clinical features. Primary symptoms include acute headache, fever, fatigue, myalgia, back pain. Lymphadenopathy is a key feature in distinguishing human monkeypox from smallpox. Kills exhaustion, a rash that can look like pimples or blisters that appears on the face, inside the mouth, and on other parts of the body, like the hands, feet, kiss, genitals, or anus. The disease typically begins with early flu-like symptoms, followed a few days later by a rash. The rash goes through different stages before healing completely. Sometimes people get a rash first, followed by other symptoms. Others only experience a rash. Diagnosis. Monkeypox can be identified by blood test or via microscope using sample swab from the rash to help indicate the presence of orthopoxvirus. Infection confirmation from patients' DNA via isolation in viral culture or PCR. ELISA test can help detect IgM indicating recent exposure and IgG indicating prior exposure or vaccination. Control of human monkeypox virus disease, treatment and vaccination. No specific treatments or vaccine. Outbreaks can be controlled by vaccination against smallpox is achieved about 85% effective prevention of monkeypox. Vaccine no longer available since discontinued following global smallpox eradication. Prior smallpox vaccination will likely result in milder disease course. Antiviral CMX001, ST246, and vaccine immunoglobulin VIG can also be used, but no data supports this. Prevention. First, reducing the risk of infection in people. In outbreaks, most significant risk factor is close contact. Raise awareness of the risk factors. Education on measures to reduce exposure to the virus. Surveillance measures, rapid identification of new cases. Public health educational message should focus on the following risks. Reduce the risk of animal to human transmission. Avoid contact with rodents and primates. Limit direct exposure to blood and meat. Thorough cooking of meat prior to consumption. Personal protective equipment should be worn while handling sick animals, infected tissues, and in slaughtering procedures. Second, reduce risk of human-to-human transmission. Avoid close contact with infected person or contaminated materials. Personal protective equipment worn while caring for ill persons. Regular hand washing after caring or visiting infected person. Isolation of patients either at home or health facilities. Following discontinuation of isolation precaution, affected individuals should avoid close contact with immunocompromised persons. Third, controlling infection in healthcare settings. Standard infection control precautions among health workers in caring for suspects and confirmed cases and their specimens. Immunization of health workers against smallpox. Older smallpox vaccine should not be administered to immunocompromised people. Suspected samples should be handled by trained staff working in suitably equipped laboratories. In transporting samples, ensure safe packaging and follow infectious substances guidelines. Clinicians, high index of suspicion when symptoms are present. Specimen collection, effective communication and precaution between specimen collection team and laboratory staff is essential. Clear labeling system for all infected samples. Proper sample collection techniques. Laboratory personnel. Laboratory exposure to poxviruses occurs primarily through needle pricks, direct contact with specimen, aerosols generated by lab procedures, limit number of staff testing specimen, personal protective equipment, rigorously applied standard precautions, avoid any procedures that could generate infectious aerosol. Veterinarians, consider all mammals as susceptible to monkeypox. Beware of animal-to-animal transmission. Ensure good hand hygiene, waste disposal, environmental sanitation, laundry. In treating suspected animals, use personal protective equipment. Protect staff, clients, and other animals. Preventing monkeypox expansion through restrictions on animal trade. Restrict or ban the movement of animal African mammals and monkey. Isolate or quarantine potentially infected animal. Quarantine all contact animal. Handle with standard precautions and observe for monkeypox symptoms for 30 days. Prognosis. Mortality rates ranging from 1 to 10% have been reported in Africa, but no fatalities occurred in the United States in 2003 and 2022 outbreak. These rates are disproportionately high in African children. Health status, comorbidities, vaccination status, and severity of complication and the amount of exposure to the virus influence the prognosis. Uncomplicated cases resolve in 2 to 4 weeks with only pox scars remaining. Deep thanks to all people in the hall attending and listening my BBT, and I hope next time attend with them. And I hope nice time in Mauritius. Bye bye. Rihab Muhammad Sudan.